I want now to give an overview over the methods used in biological spectroscopy. The range of wavelengths used in spectroscopy of biological molecules is impressive. In the high energy regime, photon energies are millions to billions of kilojoules per mole, which is more than sufficient to break the strongest covalent chemical bonds. Such high energies are used in electron microscopy and X-ray diffraction. But also photons in the UV spectral range have enough energy to break bonds and destroy molecules. In the visible and infrared spectral range, the photons have less energy than a typical covalent bond and are therefore insufficient to break bonds. Photon energy is, however, still larger than the thermal energy of 2.5 kilojoules per mole at room temperature. At the low energy end, we have microwave and radio frequency radiation. The photon energy is in the joule per mole range and thus much smaller than the thermal energy. You will recall that the ratio between photon energy and thermal energy is important for the occupancy difference between the ground state and an excited state. We have already seen that the different energy subsystems have differently large energy gaps between the states. Electronic states have large transition energies, and therefore light in the UV or visible spectral range is needed to induce them. The spectroscopic techniques in this spectral range are UV visible spectroscopy, fluorescence and circular dichroism. Smaller photon energies are required for vibrational transitions, which are observed in the infrared spectral range. Very appropriately, the technique used in this spectral range is named infrared spectroscopy. Even smaller photon energies are needed for electron and nuclear spin orientation changes. These are photons in the microwave and radio wave spectral region. All the mentioned techniques observe the absorption of photons, which means that the photon energy is equal to the energy gap between ground and excited state. Raman spectroscopy does not seem to fit into this reasoning, as it uses UV or visible light, but it observes vibrational transitions. The vibrational transition energies in Raman spectroscopy are the same as in infrared spectroscopy. So why are photons of much higher energy used in Raman spectroscopy? The reason is the following. Raman spectroscopy is not an absorption spectroscopy, where a photon is absorbed. Instead, Raman spectroscopy is based on inelastic scattering. The incident photons lose or gain some energy, and this energy difference corresponds to the vibrational transition energy. This energy difference is much smaller than the photon energy of the incident and scattered photons. Thus, in Raman spectroscopy, the energy of the used photons does not correspond to the observed transition energies, because the incident photons are not absorbed in Raman spectroscopy. All methods have their advantages and disadvantages. None tells us all what we want to know about the biological system. Each gives a specific answer to a specific question. With the answers of several methods, we can start to build up the jigsaw puzzle that represents our knowledge about the biological system. Some methods hit the molecules with a big hammer, meaning high photon energies, while others only tickle them with low photon energies. Some of the methods look at the whole molecule, some only at some part of it. This part is either intrinsic or has been introduced artificially by the researcher. Some methods can work under physiological conditions, while others require particular conditions, for example crystals or very low temperatures. What follows now is a very personal judgment of the different methods regarding their benefits and the pain imposed on the researcher. The pain factor is a measure of the effort needed and of the availability and costs of the equipment. There are three grades indicated by small letters, large letters, and large bold letters. S stands for information on structure, D for information on the dynamics of the system, and P for the pain. Electron microscopy, X-ray diffraction, and NMR spectroscopy can give structural information at atomic resolution and also information on different conformational states, even in time-resolved experiments in the case of X-ray diffraction. That's why they score high for structural and dynamical information. NMR can also be used to study the mobility of individual segments of a molecule. The pain factor is also high because very expensive equipment is needed for all three methods. In case of X-ray diffraction, researchers nowadays usually travel to a synchrotron to do the experiments. Also, X-ray crystallography needs crystals, which are difficult to obtain for some proteins. NMR spectroscopy needs quite a significant amount of sample, which can be time-consuming to produce. Electronic absorption spectroscopy works with simple lab-scale instruments and is easy to perform, but it also gives only little information on the structure of biological molecules. However, it can be used to study very fast biological processes, as the time resolution can be in the femtosecond range. This requires, of course, specialized instruments, which are often self-built. Fluorescence does not provide so much information on molecular structure, but combined with a microscope, it enables very nice studies of the distribution of molecules in biological cells. It also can give detailed information about the mobility of the studied molecules. The spectrometers are straightforward bench-top instruments, and the sensitivity so high that single molecules can be detected. The remaining methods give intermediate structural information. For example, circular dichroism and infrared spectroscopy provide information on the secondary structure of proteins. In addition, infrared spectroscopy can be used for detailed studies of the reaction mechanism of proteins with a time resolution up to femtoseconds. And EPR spectroscopy provides mobility information similar to fluorescence. For most applications of circular dichroism, Raman and infrared spectroscopy, commercial bench-top instruments can be used that are more expensive than UV visible spectrometers, but still reasonable. The sample amounts are also higher than for UV visible spectroscopy and fluorescence, but lower than for NMR spectroscopy. EPR instruments are considerably more expensive. Some methods give a lot of direct information on the biomolecule of interest. Examples are NMR and X-ray crystallography, which can reveal the 3D structure of biomolecules. Other methods are often used to monitor changes. The only requirement is a spectral probe that reports on an event like protein folding or unfolding, DNA melting, protein conformational changes, binding of ligands, and so on. This probe or reporter group can be an intrinsic group or can be introduced artificially. Examples for techniques used in this way are absorption spectroscopy, fluorescence, circular dichroism, and infrared spectroscopy. Using spectroscopy to monitor changes is a very powerful concept. It allows researchers to investigate the many parameters that affect the event of interest. Thus, a curve like that on the left-hand side gives access to a lot of important information. We take the example of ligand binding to a protein. Such type of experiments are important, for example, for drug development, where very many drug candidate molecules are tested regarding their ability to bind to a target protein. Let's assume that the protein contains a reporter group that gives, for example, a stronger signal when a ligand binds. From this it can be determined how strong a ligand binds, in other words, what concentration of ligand is needed for binding. From concentrations of free and bound ligand, the equilibrium constant and the reaction Gibbs energy of binding can be inferred. Different ligands can be compared, and in that way parts of the ligand that are important in binding determined. Mutants can be used to identify protein residues important in binding. It can be studied whether ligands bind to the same binding site or not, whether binding is affected by other parameters like temperature, pH value, and so on. Another example is protein folding. We assume that a reporter group has a different signal when the protein is folded than when it is unfolded. Thermal stability of the protein under a variety of conditions can then be probed by determining the temperature at which it unfolds. One of the questions might be how folding is influenced by mutations that abolish specific residue-residue interactions in the native structure. Protein folding and unfolding can also be followed in a time-resolved way. Then the kinetics of folding can be investigated, and whether folding is a cooperative process or whether it proceeds in a number of steps. These two examples illustrate how powerful the approach is, although no interpretation of the spectroscopic signal in terms of molecular structure is attempted. For example, in the protein unfolding experiment just discussed, it is not of interest how the structure of the reporter group and of its environment change when the protein unfolds. The spectroscopic signal is just used to monitor the process of protein unfolding without further interpretation. Nevertheless, such an approach can give molecular information, as we will see later in the course.